Good morning, everyone, and welcome. Today, we'll be diving into the fascinating world of forensic analysis, focusing specifically on hair and fiber evidence in crime investigations. We'll explore the different types of trace evidence, how they're collected, the various analytical techniques used, as well as the challenges faced by forensic experts. Plus, we'll look at the latest technological advances that are shaping the future of forensic science. I'm excited to share this comprehensive exploration with you. Let's get started. Trace evidence is essentially tiny bits of physical material that get transferred during a crime, often so small they need a microscope to be seen. These could be anything from hair and fibers to paint chips, glass fragments, or even soil. What makes trace evidence so important in forensics is its ability to link suspects, victims, and crime scenes together. Because these materials can stick around on surfaces and move between people and places during contact, they serve as a reliable way to back up other kinds of evidence. Ultimately, trace evidence plays a crucial role in piecing together what happened and guiding investigators toward the truth. Hair and fiber evidence refers to tiny microscopic materials that are often found at crime scenes. Hair, which is primarily made of keratin, is especially useful because its unique structure can help link individuals to specific locations or objects. Fibers, on the other hand, come from textiles and various materials that people wear or that exist in an environment. What makes both hair and fibers particularly important is their ability to transfer during physical contact, meaning they can connect people with places or items involved in an incident. By analyzing these small pieces of evidence, forensic experts can uncover connections that we might not see otherwise, providing crucial insights that aid the investigation. When we compare human hair to animal hair, there are some clear differences to note. Human hair usually has a fairly consistent diameter along its length, and the medulla, the central core, can vary in pattern but tends to be less complex. On the other hand, animal hair is much more diverse. The medulla structures vary widely depending on the species, and the scales on a hair's surface can tell us a lot about the particular animal. These differences are important when analyzing hair samples, especially in fields like forensic science or biology. Hair is made up of three main layers: the cuticle, the cortex, and the medulla. Starting with the cuticle, this is the outermost layer that acts as a protective barrier. It has a distinctive pattern of scales, which can vary between species and even individuals. Beneath the cuticle lies the cortex, the thickest part of the hair, where the pigment granules are located. These pigments give hair its color and can provide important clues in forensic analysis. At the center is the medulla, which is a core that sometimes may be present or absent, and its pattern can also vary. From a forensic perspective, examining these layers closely under a microscope is crucial. The scale patterns of the cuticle and the structure of the medulla can help differentiate whether the hair comes from a human or an animal and even identify specific characteristics related to the species. Meanwhile, the pigmentation in the cortex gives valuable information about the color and origin of the hair. This detailed morphological analysis allows forensic experts to effectively compare hair samples, helping them either connect a suspect to a crime or rule out involvement. When we talk about fibers, we can divide them into two main categories: natural and synthetic. Natural fibers come from plants or animals. For example, cotton is a natural fiber that comes from a cotton plant. Wool is obtained from sheep, and silk comes from silkworms. These fibers have been used for centuries in textiles due to their comfort and breathability. On the other hand, synthetic fibers are created through chemical processes in a lab. Common examples include nylon and polyester. These man-made fibers often offer durability and resistance to wrinkles and shrinking, making them popular for a variety of applications. Understanding the difference between these fiber types helps us appreciate the unique qualities they bring to fabric production. When it comes to hair and fiber evidence, proper collection and preservation are absolutely crucial. If not handled correctly, there's a real risk of contaminating or even losing valuable material, which can compromise the entire investigation. To collect these samples, we rely on specific techniques, like using clean tweezers to carefully pick up fibers, tape lifts to gather evidence from surfaces, and sometimes vacuum devices to collect particles from clothing or other items. Once collected, packaging plays a key role. Samples should be placed in well-labeled, breathable containers like paper bags or envelopes to prevent moisture buildup and mold growth. Finally, thorough documentation of where and how the evidence was collected is essential. This not only maintains the integrity of the sample, but also supports its credibility during lab analysis and any courtroom proceedings. When we talk about microscopic analysis in forensic hair and fiber examination, two main types of microscopes come into play. First, compound microscopes, which are essential for getting a detailed, magnified view of the sample's structure. This lets us observe small features that aren't visible to the naked eye. Then, we have comparison microscopes, which are incredibly useful because they allow us to look at two samples side by side at the same time. This side-by-side comparison is crucial when trying to determine if two samples might be a match. Under magnification, analysts focus on several key characteristics. These include the scale patterns on the hair, the medullary index, which refers to the central core of the hair, the color and diameter of the fibers or hairs, and any surface features that might be unique. Each of these details can provide important clues. The real forensic value comes from these visual assessments. By carefully examining and comparing these morphological traits, analysts can either link samples together or confidently exclude them. This detailed scrutiny helps build strong forensic associations, making microscopic analysis a powerful tool in investigations. When it comes to fiber identification, chemical analysis methods play a crucial role in going beyond what we can see with the naked eye. One powerful tool is Fourier transform infrared spectroscopy, or FTIR. This technique examines the molecular vibrations within a fiber, allowing us to determine its chemical makeup and identifies specific polymer signatures unique to different fibers. Another important approach is dye analysis, which looks at the chemical composition of the pigments used on the fibers. This provides extra details that help distinguish fibers that might otherwise appear very similar. Overall, these chemical methods are vital because they confirm the fiber types more accurately and help differentiate samples that look alike, greatly enhancing the strength of the evidence. Hair and fiber analysis is a vital tool in forensic investigations because it helps link suspects to crime scenes or victims through the transfer of these materials. By examining hair and fibers, forensic experts can connect an individual to a particular location or object they've come into contact with, which supports the overall investigation. For example, fibers from a suspect's clothing might match those found on a victim, or hair samples can be consistent with a specific person. This type of evidence is especially useful in corroborating witness statements, confirming a suspect's presence, and strengthening the arguments in a case. Ultimately, hair and fiber analysis helps build a clearer picture by establishing important connections between suspects and the evidence found at the crime scene. When analyzing hair and fibers, there are several key limitations and challenges to keep in mind. First, environmental degradation can affect the samples, as exposure to elements like sunlight, moisture, and chemicals can break down the material and complicate analysis. Another significant challenge is contamination during collection. If the samples aren't handled properly, they can easily become contaminated, which impacts the accuracy of the results. Additionally, there's an inherent difficulty in individualizing hair and fiber evidence. Unlike DNA, these materials often can't be linked to a single person with absolute certainty. Hair tends to show similarities among different people, and many fibers are mass-produced, so the conclusions we draw are often associative rather than definitive. This means they suggest a connection, but don't provide conclusive proof. Because of these issues, it's crucial to fully understand these limitations to interpret the results correctly. This understanding also helps us communicate the level of scientific certainty responsibly, especially in legal settings where the stakes are high. Recognizing these challenges ensures we remain transparent and accurate in our forensic work. When we look at real-life applications, forensic cases really highlight how crucial hair and fiber evidence can be. In many instances, fibers found on a suspect's clothing have directly linked them to a crime scene, providing a vital connection that might not have been possible otherwise. Similarly, hairs discovered on a victim have served as key evidence to identify and connect perpetrators to assaults. These examples clearly show the powerful role that such physical evidence plays in solving crimes and delivering justice. Recent advancements in technology have significantly improved the accuracy and resolution in analyzing trace evidence like hair and fiber. One major development is the use of digital imaging, which allows for much more detailed and precise microscopic comparisons than ever before. Alongside this, advanced spectroscopic techniques enable us to perform chemical profiling, giving deeper insights into the composition of the samples. Additionally, automated databases now assist in pattern matching, speeding up the identification process and reducing human error. Altogether, these innovations increase the precision of our analyses, cut down the time required, and broaden what we can detect, making forensic examinations of hair and fiber more reliable and comprehensive than in the past. Hair and fiber evidence play a crucial role in forensic investigations because they have a unique ability to connect people and places through physical transfer. By understanding the different types of hair and fibers, their structures, and how to properly collect and analyze them, forensic professionals can uncover valuable information that might otherwise be missed. While there are certainly challenges and limitations in working with this kind of evidence, ongoing technological advancements continue to improve our ability to analyze these materials more accurately and efficiently. Ultimately, hair and fiber evidence remain indispensable tools in pursuit of justice and help ensure that crime scenes are accurately reconstructed.